and then following by Jiaying, and the last one of the session will be Laura. Welcome everyone to our first Global Science Exchange series. And today's topic is Wastewater Surveillance of SARS-CoV-2. And we have 3 speakers today in this first session. They are Christine, Jiaying, and Laura. So let me introduce our first speaker to everyone. This is Christine Kandowski. Apologize if I incorrectly pronounced the name. Christine is a Ph.D. student in the Environmental System graduate group at the University of California, Merced. She joined Dr. Colin Norton's Food Energy Water System for and with the Underserved Research Group in the summer of 2021 to aid the Healthy Central Valley Together program, the COVIDPOOPS19 dashboard of the wastewater monitoring locations, and the wastewater SARS Public Health Environment Response Global Data Center projects. Her background is in civil and environmental engineering with 4 years of water and wastewater sector experience. So let's welcome Christine to give us her first presentation.
Thank you very much. Let me share my screen. Can everyone see my screen okay? Okay, so hello everyone. Thank you for having me as a speaker for the ASCE Global Science Exchange. My name is Kristen Kudonsky and I'm a first-year PhD student at the University of California, Merced in the Environmental Systems program. My advisor is Dr. Colleen Naughton, and I'm a part of the Food, Energy, Water Systems for the Underserved Research Lab. And today I'll be presenting to you on my research on data equity and analysis of global access to wastewater monitoring for SARS-CoV-2. And so today I'll cover an introduction on wastewater-based epidemiology, and then go into my project work on the COVIDPOOPS19 dashboard, as well as the WSphere Global Data Center, and then dig a little deeper into the equity analysis that I've been doing with the data from both projects, and then acknowledgements and questions. And you can see a picture of our lab group on the screen as well. So as you know, we have been experiencing the COVID-19 pandemic since early 2020. Individuals infected by the virus primarily suffer from respiratory symptoms, hence why we all wear masks. But there is also a gastrointestinal manifestation of the disease, which leads to fecal shedding of SARS-CoV-2 RNA into our sewer systems. And fecal shedding has not been found to be infectious. And wastewater-based epidemiology, or wastewater surveillance, is the tool used to measure pathogenic organisms, biochemical markers, pharmaceuticals, and markers of antimicrobial resistance within the sewer catchment area of a community. It has been used for the past 2 years to monitor the concentration levels of SARS-CoV-2 RNA. And this diagram shows the workflow of the collection process as well as the lab process. And so individuals from a residential or commercial building will shed the virus into the wastewater collection system. And then shown here are 3 examples of sample locations, although you can sample from a non-sewered area as well. And the 3 locations on the screen are the maintenance hole or manhole, the pump station or lift station, as well as the wastewater treatment facility. And so once the sample is collected, it is then sent to the lab for the solids to be settled and then prepared for extraction. And then SARS-CoV-2 RNA is extracted from the sample and then set into an RT-qPCR, which is the real-time quantitative polymersion chain reaction to detect the concentration levels of SARS-CoV-2 RNA. Wastewater-based epidemiology is a supplemental tool to public health surveillance or clinical testing, and where one wastewater treatment plant can provide data on the health of a population within a community and represents asymptomatic as well as symptomatic cases, compared to clinical testing where one clinical sample is indicated for one infected individual. In addition, infected individuals shed the virus several days before the onset of symptoms. Therefore, the virus is detected in wastewater before patients receive clinical swab testing. And so as wastewater-based epidemiology became more popular and used as a tool for SARS-CoV-2 within communities, our research team decided to start tracking where wastewater monitoring was being implemented around the world. And we do this by keyword searches on social media, such as Twitter, or attending various webinars, or scientific literature and internet searches, as well as reviewing the submissions to our Google Forms. And so all of this data is categorized by myself and a team of students into categories of dashboards, universities, countries, and sites. And as you can see, we have 113 dashboards, 276 universities, 58 countries, and over 3,000 sites. And this process is continuously updated on a weekly basis to an ArcGIS story map, and that is our COVIDPOOPS19 dashboard. So from collecting this data, and as you can see on the map, there's some areas where wastewater monitoring is missing or lacking. And so we found that primarily in high income countries, or at 65%, wastewater monitoring is being implemented, and there's less access to it in low and middle income countries at 35%. And so data is also not widely shared publicly or accessible to researchers to further inform public health responses. And then in addition, just from the dashboards that I've reviewed and looked at, not all of them have data that's available to be downloaded and used for research. And then you can also read more about this in the Shows to Data preprint. And so the COVIDPOOPS19 project then leads into another project I've been working on, which is the Wastewater Sphere Global Data Center in partnership with the Global Water Pathogens Project. And this project is in collaboration with Michigan State University, UC Merced, PATH, Benthic, as well as KWR Water Research Institute in the Netherlands. And so as you can see, WSphere stands for Wastewater SARS Public Health Environmental Response. And WSphere expands upon the dashboards that we collect from COVIDPOOPS19, and especially the ones with downloadable data, to showcase the trending data of presence and absence and the sampling data of SARS-CoV-2 concentration levels. In addition to the global map, there are use cases that showcase examples of how wastewater data has been used to support public health actions in different settings, such as national, regional, local, building level surveillance, high and low resource settings, sewered and non-sewered settings as well. And the three use cases that we have are currently in Switzerland, the Netherlands, and Catalonia, Spain. And so as I mentioned, dashboards are curated from COVIDPOOPS, and the ones with publicly accessible downloadable data is then moved into our data collection phase. And then dashboards that do not have publicly accessible data, or dashboards that are provided to us from data providers, are then, we then request to arrange a meeting with them and sign a data agreement to be allowed to use that data on our website. And so once data collection has been finished, then it is assigned to either myself or other graduate students or postdoc students to conduct a data review. And so during the data review, we're just looking for any missing or additional information that we need for a complete data set to be put on the website. And so typically missing data might include SARS-CoV-2 concentration levels. So we'll see data that has presence and absence, but not necessarily the SARS-CoV-2 concentration levels. And that information is highly valuable to understanding the behavior of the virus. In addition, we might not have the gene targets that were used or the lab methodology. And so all that information can help us understand and track the virus better. And so then once we have that information, we import it into a repository, a final data review is conducted, and then it is published to our map. And so here is a screenshot of our current global map. It has 100 dashboards, 16 published data sets, and 26 countries. And then at the top bar, you can see the tab for the global map, and next to that is the data tab, which is where you can access our downloadable data for all of the information that we have. And next to that is the use cases that I mentioned before. There's an About tab, and then also the Contribution tab, to where anyone interested in contributing their monitoring data can submit it to us, and we'll reach out for an initial meeting and then also start reviewing the data. And so further looking at the data, we have over 1,000 monitored sites, almost 3,600 samples, 8 lab methods, and then 3 use cases. And then For site types or sampling locations, as I mentioned before in the wastewater-based epidemiology diagram, sampling locations are wastewater treatment plants, sewers, open sewers, and then non-sewered areas such as rivers or beaches. Our gene targets that we typically see are N1, N2, and E, or combination, and the combinations are usually N1 and N2 together. We sample from raw sewage as well as primary sludge, and then a monitoring scale of building, urban, regional, and national. And the one building level dashboard that we have is from New Mexico. On average, our sampling starts in fall of 2020 from our dashboards. However, in low and middle income countries, I've noticed that sampling occurred typically in spring of 2021. And so some of the countries that we have listed as well, and then the low and middle income countries that we include, such as Malawi, Nepal, and Pakistan. And so coming soon to 2022, this year, we hope to expand the global data center to 1,500 sites, 90,000 samples, 30 lab methods, as well as 6 use cases, and adding an additional 15 countries, and 5 of those being low and middle income countries, such as Bangalore, Brazil, Ghana, Indonesia, and South Africa. And so with this data, and a part of my research is I'm developing a systems model for 2 case studies, one in a high income country and the other in a low and middle income country, to compare the different variable parameters that influence SARS-CoV-2 behavior in wastewater. And so these variable parameters are based on key events which occur in each country. And it can include when vaccines became available, when pharmaceutical treatments became available, or seasonal events such as holidays or school. And that can also include gatherings such as weddings or funerals, mask mandates, shelter-in-place orders, illicit or licit drug use, as well as fecal shedding rates. And then future considerations are variants of concern, as well as transmissivity rates. And so for some of my preliminary data, the high-income country that I was considering is Scotland. And so on the right-hand side, you can see screenshots from the Global Data Center. The top screenshot is the sampling tab, where you see the presence and absence of SARS-CoV-2 within each community. And then below that is the trending data, which shows whether or not the SARS-CoV-2 concentration is increasing or decreasing. And then on the left-hand side, on the top figure is the SARS-CoV-2 RNA concentration data within wastewater from the samples collected in East Calder, Scotland. And below that is the clinical data from the Johns Hopkins University COVID-19 dashboard. And so the wastewater concentrations of SARS-CoV-2 began to spike at the end of August 2021, while the clinical case data began to spike a few weeks later in early September 2021. So further showing that early detection of virus within the community before clinical symptoms started to occur or clinical testing. And then additionally, the Scottish school system typically begins in the 2nd or 3rd week of August, which translates to the timing of the spike in SARS-CoV-2 concentration levels within the community, as indicated by the red arrows. And so for the low and middle income country that I selected is Nepal, and then specifically the city of Kathmandu in Nepal. And then similarly, last to the slide, on the right-hand side are screenshots of the global data center, the top being the samples view and the bottom being the trending view. And then on the left-hand side is SARS-CoV-2 concentration taken from samples in the sewer shed and not at the treatment plant. And then below that is the clinical case data from Johns Hopkins University from the COVID-19 dashboard. And so I want you to notice how the quantity of wastewater sampling data from Scotland to Nepal, Scotland's monitoring was relatively even, occurring from December 2020 to November 2021, while Nepal has sparse wastewater data points beginning from March 2021 to October 2021. There is a lag in monitoring start time and a lack of sampling data, and this makes it difficult to evaluate a comparison between the wastewater and clinical data. You can see there's a single spike in SARS-CoV-2 RNA concentration at the end of June 2021, and from a single sample collected. And the remaining concentrations drop off while the clinical data shows an increase throughout the months of July and August. So this may indicate less robust monitoring in Nepal and negatively impacts the public health response in communities lacking wastewater monitoring and data, which further highlights the importance of establishing wastewater monitoring programs and providing equitable resources to maintain wastewater monitoring programs within every community. And so for both data providers, they will be contacted before any of this data is being published. And so I would also like to acknowledge previous and current students who have worked on these projects or are working on these projects with me, and then also mention funding from Citrus and then our collaborators. And so thank you for your time. I'm Kristen Kadonsky. You can contact me at katekadonsky@ucmerced.edu, and you can follow us at COVIDPOOPS19 on Twitter, or visit the WSphere website at www.sphere.waterpathogens.org. And so thank you for your time.
Mara, for the questions, and thanks Christine for the awesome presentation. So let's move on to Jiaying. So Jiaying, Dr. Jiaying Li is a postdoc research fellow at the Queensland Alliance for Environmental Health Science, the University of Queensland. She obtained her Ph.D. in 2020 at Advanced Water Management Centre, University of Queensland, which was focused on understanding transport and transformation of biomarker in solar systems. She is now involved in the Queensland's COVID-19 wastewater surveillance program. Her research interests include source tracking of emerging contaminants in solar networks, understanding and modeling the fate of biomarkers in urban water systems, and systematic monitoring of biochemical pollutants in the environment. So let's welcome Jiaying to give us her presentation.
Yeah, thanks Yitian for the introduction. And since my network is not quite stable, so forgive me if there is any interruption. Can you see my screen now?
Yes.
Okay, cool. So it's my pleasure to be invited here and present my works with people around the world. So the title of my presentation today is Tracing Virus in Wastewater to Monitor COVID-19 Spread in Communities. As Christine has presented in the previous presentation, monitoring SARS-CoV-2 in wastewater is now recognized as an important environmental surveillance to complement the clinical-based surveillance, because the virus RNA could be shed from both symptomatic and asymptomatic cases, and the shedding starts from pre-symptomatic stage before people go for clinic testing. So this indicates that the wastewater monitoring will be able to provide an early warning of COVID-19 incidents in the population. And usually we collect samples at the inlet of a wastewater treatment plant, which receive wastewater from the entire catchment. So we will be able to interpret the COVID-19 prevalence at the catchment scale. So in the past 2 years, the COVID-19 wastewater surveillance program has been developed and implemented in Australia and over the world. So when we are designing and developing the program, we encounter many questions and challenges. And one of the important questions that we are facing is like, when we detect positive signals at the wastewater treatment plant, where did it come from? Like, is it from a quarantine site where infected people are under quarantine, like quarantine hotels and quarantine hospital, or it is from local community, which indicate the potential community transmission? And if it is from the local community, where is the origin of the infected people and infected cluster? And this information is important to help the local public unit to make rapid response. And we realized that if we purely rely on the wastewater monitoring at the wastewater treatment plant, we won't be able to know where they come from. So because the wastewater treatment plant receive wastewater from the entire catchment. So in order to better understand the potential community transmission, it required us to shift wastewater monitoring from wastewater treatment plant to its upstream catchment, which is the sewer systems. So when we are trying to transit from wastewater treatment plant to its upstream catchment, the first question is, how do we take samples from the sewer system? Working in the field means you don't have too many access to power, and it's not feasible to use expensive facilities at multiple sites. You won't be able to manage them. So what we are using for the field monitoring is something called passive sampler named Torpedo. This is a sampler that contains the electronegative membranes in a 3D printed housing unit, which is designed by Professor David McCarthy at Monash University, Australia. So this is a very simple sampler, which is has 15 centimeters long, and it's easily to be deployed in the sewer manhole. Okay, so our previous study demonstrated that the membrane in these samplers is able to perform time-integrative sampling, which means the membrane can continuously capture and accumulate virus from wastewater over 48 hours. It doesn't require external power or something. It just continuously uptake, capture and accumulate virus. Okay, so the next question is, where should we deploy these samplers in the sewer system? Similar to the road networks, sewer system is another joint system underground. So you can see this kind of manhole everywhere on the road, on the street, and in a city-wide catchment, there could be thousands of such manholes. So where are the optimal, the representative manholes that we should take samples from? This is an important question for us. So each manhole serves like a sub-catchment. Apparently, the sub-catchment couldn't be too large, because it means that if you detect a signal, you still don't know where they come from. And it couldn't be like everywhere, because if it serves a very small catchment and it's everywhere, you won't be able to collect all of them in one day. Like if there are more than 20, 30 manholes in the city, it's not possible to collect them in one day. So ideally, we would like to select the manholes, this representative manholes that service specific, separate individual subcatchments. And ideally could be 10 to 20 manholes, which allowed us to deploy and retrieve these samples in one day. So the next question is, if there is any 1 infected cases or less than 5 infected cases living in this subcatchment, is the passive sampler sensitive enough to capture the signals from these infected cases? So this is the same question like, what is the lowest infection rate in the community that allows for the detection by the passive sampler? Therefore, we designed a study to track the SARS-CoV-2 RNA in sewer networks. The aim of this study is to determine the sensitivity of passive samplers for COVID-19 incidents in the community. So the right figure shows a subcatchment of a city, and there is a quarantine hospital in this subcatchment. This quarantine hospital is considered as the single known source of SARS-CoV-2 in the catchment that contribute SARS-CoV-2 RNA into the wastewater, because there were no local cases were reported during our study. So we select 8 manholes along the trunk sewer, the main trunk sewer, downstream of this quarantine hospital, and the service area of each manhole increased from upstream to downstream. So we conducted resampling campaigns in this study, and there were 13 to 25 COVID-19 patients in hospitalization. The according infection rates range from 0.8 to 18.6 per 10,000 people, and the infection rates decrease from upstream to downstream when there are more and more people being served by a certain manhole. And in this study, we deployed Torpedo for 24-hour sampling. So we deploy 17 samplers in total in this study, and we detect the positive signals of SARS-CoV-2 from 15 of them. And the samplers were, the signals were not detectable at the last 2 sampling sites. The number of RNA on membranes decreased along the silver networks with increasing dilution, and we found a positive relationship between the numbers of SARS-CoV-2 RNA and the infection rates in the silver shed. And we found that the lowest infection rate that allow for the positive detection by the membrane is around 2 cases per 10,000 people. When the infection rate is below 2 cases per 10,000 people, the membrane in the passive samplers is not able to capture any signals from the wastewater. With an enhanced understanding of the sensitivity and ability of the passive samplers, we used them in our upstream Sentinel silver sampling program in the 2 urban catchments in Brisbane City, catchment A and B. So we built 11 sampling sites in these two catchment, and there are 11 specific individual subcatchments for the upstream monitoring. In catchment 1, there are several quarantine hotels and hospitals located in the mid of the catchment. So most of the this upstream monitoring site located at very upstream of the catchment. So during April to November 2021, we conducted daily or weekly sampling event in the catchment. And this Brisbane City was under well control. There was no local transmission occurred in the city until the end of July 2021. The first public health alert of a local transmission was reported, and the outbreak occurred at a local school, which is located at the boundary of catchment A and B. And you can see that there are 2 upstream monitoring areas service the suburb and areas around the locations of this local school. So this is the zoom-in map of those 2 subcatchment A1 and B1, and this black arrow indicates the timeline of the cluster depending on the clinical data. So in at the end of July, the first public health alert was announced, and the city ended lockdown. The lockdown was lifted nearly 1 week after, and then the last infected cases related to this cluster was reported near the end of August. The below heat map represents the timeline based on the wastewater data, and you can see the first positive signal in wastewater was detected from A1 1 week ahead of the clinical confirmation, and the positive SARS-CoV signals was continuously being detected in A1 and B1 until 2 weeks after the final clinical case was reported, and the highest level of SARS-CoV-2 wastewater occur in the few days before and after the lockdown lifted. So in total, the positive signals in wastewater in A1 and B1 lasted for 7 weeks, which is longer than the 3-week clinical record. In conclusion, our field scale study and monitoring demonstrate that dilution will importantly affect the signals of SARS-CoV-2 in sewer system, and passive sampling is a simple and practical tool to monitor the spread of virus in upstream subcatchments, and it will allow us to monitor potential community transmission before the clinical confirmation, and the sensitivity of torpedo passive samplers for COVID-19 incidents is around 2 cases per 10,000 people. This is an important parameter for site selection, and upstream sampling can identify hotspots, provide early warning and reflect the trend of disease prevalence at the community level. So that's it, the end of my presentation, and I will acknowledge University of Queensland, CSIRO, Queensland Health and Urban Utilities for their support. Thank you.
Thanks, Jiaying, for the beautiful presentations. Any questions from the audience or other presenters? Feel free to ask questions. We still have 5 minutes for Jiaying sessions to accept questions. If not, I'll probably ask the first one, and then someone else with questions can come up later. So Jiaying, if you don't mind, can you please explain how did you get the figure you can detect 2 in 10,000, like 2 cases in 10,000 populations?
Yep, it's let me go back to the figure. So here is the figure. So during our, oh, sorry, where it is? Okay, during our study, the infection rates range from 0.8 to 18.9 in cases per 10,000 people. And each infection rate is has a specific concentration level that of SARS-CoV-2 that we detected from the membrane. And for the sampler, that it has a corresponding infection rate of 2 cases per 10,000 people. That is the final sampler that we can detect the positive signals of SARS-CoV-2 from. And for the other 2 samplers where the infection rate were below 2 cases per 10,000 people, like their corresponding infection rate were 0.8 and 1.9 per 10,000 people, we will not be able to detect any signals from those samplers. So this suggests that 2 cases per 10,000 people could be the lowest infection rate that allow us to detect signals from the membrane.
All right, thanks for the explanations. And also I have one question. I believe in the first study, and you used the quarantine hotel as the source of viral contaminants. So in the next study, like the study built on the first study, and you want to actually detect how many cases can, how many cases.
I want to detect if there is any community transmission in each subcatchment.
Yes. So in the second part, did you put multiple samples along the source chain?
The second study?
Yes, the second study.
And they are separate. There are different study designs. So this study, we aim to monitor if there is any potential infection, people living on traveling in each subcatchments. So we deploy samplers at every manholes that service this specific subcatchment. So they are not in the same line, they service independent subcatchment in this study. And they are all located upstream of the quarantine hotel and hospital in order to exclude any potential contribution from these sites to the wastewater. Yes.
So each catchment has different characteristics. You can see the geometry shape is different, and I believe the land use characteristic is different as well. So how did you manage to to adjust for the difference between each catchment to make the comparison later on?
So we are trying to see for the 11 samplers that are deployed at these sites every day, we want to see if they if we can detect any signals from them. If not, that's good. That means there's no infected people in this catchment. If we do detect any signals from this catchment, that it means that something happened in this subcatchment, and we reported it to the public health unit. So in terms of comparison, it's like something like a simple yes or no comparison. Yeah.
All right, thanks, Jiaying, for the beautiful answer. I'm looking forward to Laura's presentation. So let me introduce Laura to all of you.
Yitian, there are two questions in the message box. I'm sorry.
Okay, sorry,
check them?
So question from Shailesh Agarwal. And the question is, nice application of passive sampler. Did you try finding association between sensitivity versus limitation due to the flow rate and the overall dilution effect? For example, what was the flow rate at a sampling point where you could detect during 2 cases scenario?
Okay, this is a very good question. So in this map, you can see, so this subcatchment is a residential catchment, and this little figure you can see the population and the flow rate is like they are well corresponding to each other. So all the flow rate is associated with people activities. So at N7, the flow rate is about 50 liters per second. And at N7, the service population is nearly 200,000 people. So, so we would suggest that the signals will be diluted from upstream to downstream, especially at the inlet of wastewater treatment plant. And this is the association that we could determine from the study, that when the flow rate at N7, the flow rate is nearly 500 liters per second. But the case in this study is that we know the number of infected people in this subcatchment, like there are 13 to 25 people living in this catchment. So this correlation, I'm not sure whether it can be transferable to other cases, because at a site with higher or lower sampling flow rate, the ability of the passive sampler to detect signal is dependent on how many infection cases in the total population serviced by this manhole. Yeah. So what we are trying to see is when there are the conclusion of our study is like when there are nearly two cases among 10,000 people, this is the limitation or the best sensitivity of the membranes to capture the signals from the wastewater.
All right. Another quick question. What gene targets are you monitoring for? The question is from Colleen Norton.
The gene targets, I think it's SARS-CoV-2. Yeah, because I'm mostly doing the field work and sampling work. Our samples were analyzed by another lab, but I believe N1, N2, I think the lab used both N1 and N2. Yeah.
All right. So quick question from Shailesh, because he or she has raised his or her hands.
Yes.
So it's me, it's he, and just to make a brief question to that, can you tell me specifically the flow rate for the sampling point where you found 2 cases? Just to get an idea.
And so the 2 cases, like it's from, I think from sampling N5, so the sampling rate is about 150 liters per second in the trunk sewer.
Okay, thank you.
Yeah. So an important point for the deployment of this torpedo passive sampler is that it requires continuous flow rate. So our previous test we found that when we deployed it in the pump station where there is no continuous flow rate, the passive sampler, it doesn't have a good performance when it's deployed in the pump station with like stagnant wastewater for a while. So it's better to deploy it in the functional gravity sewer with continuous flow. Yeah, I see a question from the audience. Is the torpedo sampler commercially available? Yes, it's designed by Professor David McCarthy at Monash University. So you can contact Professor David for this torpedo sampler.
All right.
Yeah.
So
you're from David McCarthy's group.
Laura. So Laura is working at the Technical University of Darmstadt. Yes. And she's working together with Professor Suzanne Lechner and Dr. Shailesh Agarwal, who just raised the beautiful questions, and working on SARS-CoV-2 monitoring in wastewater. Other focuses are antibiotic resistance genes and pathogens, as well as understanding microbial communities in nitrogen removal system. All right, let's welcome Laura.
Hi. Can you see my screen?
Yes, I can see it.
Okay, perfect. So then, good morning, good afternoon, and good evening. My name is Laura Orschler, and I'm working together with Susanne Lechner and Shailesh Agarwal at the Technical University in Darmstadt. And today I want to present you our work of wastewater surveillance for SARS-CoV-2. So when we think about wastewater, or this was what happened most to me, people are thinking mostly about sewer bills and the smell and toilets and so on. But wastewater has so much more to offer. We can regain a lot of nutrients. And wastewater itself is kind of the mirror of society, of a specific area or a city, because for example, we can see what drugs are taken in that area, or we can see what are the most antibiotics used. And this is also why we can use wastewater, or the information we can get from wastewater, to understand more about the pandemic. Wastewater surveillance itself is actually nothing new. It's known before then and was mainly performed for polioviruses or noroviruses. And with these studies and the information that were already there, we were able to establish the wastewater surveillance for SARS-CoV-2 very fast, because already in March there were several studies that published information for detecting SARS-CoV-2 in wastewater. And the question is, why does it make sense to do wastewater testing? And it's different in every country, I think. So I will focus more on Germany, because this is really, or this is mainly special. In Germany, the tests are regulated somehow, so that at some point you have to get tested to do specific things, especially if incidences are very high, or you're not vaccinated, or so on. Or you are just interested to know, or you are careful and you want to know, are you positive or not? And this also means that the more tests I have, as you can see here, the probability that I get more positive tested people increases. But this makes it also a little bit difficult. It's kind of a challenge, because I cannot, I'm always behind the distribution of the pandemic. And this is not only for the number of tests, this is also the case for the proportion of whole genome sequencing, because it's unfortunately in German, but the gray bars are the number of COVID-19 cases, and the red line is the number of tests that are chosen for genome sequencing. And here you can see the light blue are suspected cases for Omicron in the different states of Germany, for example, North Rhine-Westphalia. And we have around 35,000 suspected cases and only roughly 4,000 confirmed cases. And this shows how the clinical data is good, but especially in the case of Germany, we do need more information to understand the pandemic better and be able to handle the situation in a different way. And there are a lot of studies since the beginning of the pandemic. As you can see here, there was a high interest of media in Germany, and there are different study groups working on different topics in this case. So now I want to go more into detail about what we can get out of these wastewater information. The information that we can generate are on the one hand trends, meaning how is the concentration of SARS-CoV-2 changing in the wastewater over time, and also the detection of mutations. And therefore, I think you've heard that before, there are two different methods. On the one hand, we have the quantitative detection with qPCR, and on the other hand, there is the detection for mutations or VOCs with sequencing method. It's mainly the same what we have for the human samples. And now I'm going to introduce to you the method, how it works. So we get samples from the wastewater treatment plant. This is either a 24 or 2-hour composite sample or a prep sample. Then there are different volumes that are either centrifuged via ultracentrifugation, or we filter it through an electronegative membrane. This is necessary because we have to concentrate the sample before we can move on with further steps. And then there's the step of RNA extraction. This is possible either by bead beating or magnetic beads. And then again, we have the two methods that we can use for the analysis, the quantitative detection or the genome information. And in the, when we started or when we moved on last year, we decided to focus more on sequencing because in Germany, there are a lot of groups working on the quantitative aspect, and our focus is more on detecting mutations. And this is why I'm going to dive more into the sequencing part here. So as I said, the method for sequencing is the same for wastewater samples than for the human samples. The only difference is that in wastewater, we do get a lot more different snippets of different variants, which makes it really hard in the end after RNA extraction and amplification to glue it together in the end and to see what different VOCs or mutations do I find in wastewater. And how is that done? Therefore, I have the genome sequence of the original Wuhan strain. And whenever there is a change in the nucleotide position, then I found a mutation. And this is similar to when we do have different variants, because here in this case, we have a combination of different mutations in the genome. And then it depends on, is it a variant of concern, where you can see it has an increase in virulence and all these different things, or it's a variant of interest, or maybe it's a variant that hasn't been found before. And these are the answers that have to, the question that have to be answered. And our workflow is that we do get the wastewater sample, we perform ampli-seq sequencing, and then we compare our results with the reference sequence for mutations, and then we report the mutations for the wastewater samples. And with this method, we were able to detect the Omicron variant in Germany already in the before the first confirmed clinical report from one passenger that flew by, and that shows how powerful this method actually is and how it could support the information, the clinical data information. And this is also what ECDC said, that we should use these sequencing methods of SARS-CoV-2 from wastewater as a complement to other monitoring methods. And this is something that we are working on, because we think that can be really helpful information to all other clinical data that we have. And now I also have an overview about another work that we have here in Germany. Darmstadt is in the state Hesse, and in Hesse we do have a countrywide sequencing project. Here you can see 18 different wastewater treatment plants, and we are monitoring them since the September 2021. And in the beginning, we could only detect the Delta variant over the time, and since the middle of December, we found Omicron here. And in one wastewater treatment plant, it was also end of November, where we did detect the Omicron variant, even when it was not that prominent in Germany by then. So for some, we don't have data from this year, but we will get that data. But for everything else, since December, we did find Omicron already, which is really good and important information for the distribution of the Omicron variant. And this leads me to the conclusion that we do have really good ways to do wastewater monitoring, both with PCR and sequencing methods. When we want to have a closer look onto the VOC, it's a little bit difficult to use qPCR with that. And therefore we recommend genome sequencing, because here we can get a better overview of the mutations and the different variants. And in case of Germany, the next steps are that we have a pilot implementation in Germany. So there will be a German-wide wastewater monitoring based on the EU recommendation that will start in February, and we will do the sequencing part in that project. And the goal would be to get structures for the future where we can use that for other pathogens, or for more detailed wastewater-based monitoring. And then I want to thank the group and all the fundings. And yeah, if you have some questions, let me know.
All right, thanks, Laura. We're finishing up the presentation finally. I apologize for all those disruptions that's happening to everyone who present and listening. Any questions from the audience?
I think there's one question in the chat.
Yep, there's one question in the chat from Colleen.
Yeah.
Norton. And she is asking, are you monitoring for BA2?
We do that, but right now I don't have any actual data on that, but we do distribute between BA1 and BA2.
Another questions in the chat. The Colin, she is asking, at the airport, did the treatment plan only cover the airport or wider city too?
We get 2 samples in that case for the transport hub, like the airport, and then we also take a sample from the city wastewater treatment plant. So we do cover both actually.
Can I ask a quick question, Lara?
Yes.
So what kind of team members you have? Because this is very like sequencing has more really lab work and pure science. Are there different expertise in your team? Can you please give us a little bit information?
I think that we are working with an AmpliSeq panel that's provided by the company Thermo Fisher, and they do have really good automated systems by now. So there's a lot of stuff that can be done with automated systems. For example, the RNA extraction, or if we do the the template preparation. I think the most crucial part for sequencing is really the bioinformatic analysis, because you really have to be a little bit in the topic and work with R to be able to analyze your data. Otherwise, I think it's a little bit difficult to do the analysis.